All right, welcome to this explainer on the epic biochemical journey of how plants leaped from water to land, fundamentally transforming our planet forever. Today, we're going to look past the macroscopic leaves and stems you see outside your window, and instead, we're diving deep into the critical, complex molecular adaptations that made terrestrial life possible in the first place.
So I want you to really challenge yourself for a second and imagine a completely lifeless, barren Earth. Because before plants made the leap, the land was just a hostile void. Think about the monumental physical challenges early life faced when trying to survive outside the comforting, buoyant embrace of the water. It wasn't just a change of scenery, you know? It required a literal, fundamental molecular re-engineering of life itself.
Here's our roadmap for today. First, we'll look at the barren terrestrial frontier. Second, the molecular clues and algal ancestors. Third, we'll break down the terrestrial survival toolkit. Fourth, we'll analyze some hard data on how bryophytes altered the climate. And finally, the vascular plant revolution.
Section 1. The barren terrestrial frontier, facing the void. To truly appreciate plant biology, you have to picture the Earth about 1.2 billion years ago. No forests, no grasses, no soil as we know it today. The land was just a thin, incredibly vulnerable coating of cyanobacteria and protists, basically just clinging to the wet edges of a barren rock. Any organism attempting to leave the water faced a brutal, dual threat. First, there's the constant, fatal risk of desiccation, drying out rapidly under unfiltered sunlight in an atmosphere that completely lacks the moisture they evolved in. Second, gravity. Water naturally provides buoyancy. If you take a soft-bodied aquatic organism out of the water, it just sags and collapses under its own weight. Overcoming these two massive physical barriers required some seriously incredible biochemistry.
Section 2. Molecular clues and algae ancestors, finding the missing link.
So, who were the evolutionary pioneers that first managed to solve these brutal terrestrial problems? Well, we find the smoking gun in precise cellular evidence that links the ancestors of land plants to modern xerophyte green algae, organisms like Zygnema and Coleochaete. And we aren't just looking at superficial similarities here, we're looking at the exact molecular machinery. Both land plants and xerophytes share distinctive circular rings of proteins embedded right in the plasma membrane that synthesize the cellulose microfibrils of the cell wall. Other algae have linear sets, not rings. Furthermore, they share highly specific details in cell division, namely the formation of a phragmoplast, which is a specialized group of microtubules that helps build a new crosswall between dividing cells.
But, as you know, just being related isn't enough. You actually need the functional tools to survive the transition. Enter a true biochemical marvel, sporopollenin. This is a highly durable polymer that acts as the ultimate chemical armor. In charphytes, it protects zygotes from occasional drying. But as plants moved to land, this exact same polymer encased early plant spores, allowing them to be safely dispersed through dry, harsh air without suffering fatal desiccation. It was absolutely crucial. It's the molecular shield that opened up the terrestrial frontier.
Section 3. The Terrestrial Survival Toolkit, Engineering Life on Land.
Now, to survive permanently above the waterline, early plants developed a fascinating and complex reproductive strategy, the alternation of generations. You've likely encountered this before, but the critical thing to really grasp here is the presence of both a multicellular haploid and a multicellular diploid organism operating within the same life cycle. A multicellular haploid gametophyte produces gametes. Those fuse to make a diploid zygote. But instead of just immediately undergoing meiosis like some algae do, this zygote divides by mitosis to become a whole new multicellular organism, the diploid sporophyte. This sporophyte then produces the haploid spores through meiosis, which eventually grow back into the gametophyte. This two-stage cycle is a game changer. It allows for incredible genetic diversity and the mass production of those heavily protected spores we just talked about.
And part of this survival toolkit involves actively taking care of the next generation. Let's zoom in on the cellular level for a second. True plants are called embryophytes because they actually retain their multicellular embryos within the tissues of the female parent. Much like in mammals, maternal plant tissues protect and actively feed the developing plant embryo via these crucial specialized placental transfer cells. These cells feature elaborate ingrowths in their walls to maximize surface area, effectively pumping sugars and amino acids directly into the vulnerable embryo.
Section 4. Data. Bryophytes alter the climate. A global cooling mystery.
Okay, let's zoom out from the molecular scale to analyze some hard empirical data on the massive ecological impact of these early plants, the bryophytes. These non-vascular plants, things like mosses and liverworts, didn't just survive on land, they fundamentally altered the Earth's climate. The data brilliantly illustrates the link between early plants and Ordovician global cooling. For example, researchers placed different rocks in water with and without macerated moss. If we look at the results for andesite rock and calcium ions, the control water released 1.54 micromoles. But with the experimental moss added, that number jumped dramatically to 5.46 micromoles. Even without true roots, these early non-vascular plants significantly increase the chemical weathering of minerals, and that process pulls carbon dioxide directly out of the atmosphere, reacting it with the released calcium to form marine rocks. Basically, these tiny plants literally scrubbed CO2 from the sky, directly contributing to a global cooling event and widespread glaciations.
Further data analysis reveals how bryophytes transform sterile ground into nutrient-rich soil over time. Tracking the feather moss pleuroseum in recovering forests shows something amazing. At 35 years after a fire, nitrogen fixation is a mere 0.001 kilograms per hectare. But as the moss establishes a symbiotic relationship with nitrogen-fixing cyanobacteria, look at the rate after 300 years. It absolutely skyrockets to 3.0. These plants functioned as molecular factories, aggressively pulling inert nitrogen from the air and locking it straight into the terrestrial ecosystem, which completely paved the way for larger plants to follow.
Section 5. The Vascular Plant Revolution, Defying Gravity.
So, while early bryophytes successfully altered the soil and the atmosphere, they were severely limited by physics. They were strictly ground-hugging, rarely growing more than a few centimeters tall. To conquer the skies, plants needed a massive molecular upgrade. Contrast those severe size limitations against the competitive potential of vascular plants. Bryophytes depend entirely on simple diffusion to move water, which keeps them permanently tied to moist surfaces. Vascular plants, on the other hand, evolved an internal plumbing system. They developed the xylem to carry water and minerals up from the roots and the phloem to distribute sugars down from the leaves. And that unlocked towering vertical growth.
But as you can probably guess, internal plumbing alone doesn't solve the gravity problem. You need strong pipes. Enter lignin. Lignin is a highly complex, incredibly tough polymer that chemically reinforces the cell walls of water-conducting xylem cells. This was the ultimate biochemical structural reinforcement. Lignin provided the rigid mechanical support required for plant stems to finally defy gravity, outcompete their neighbors for sunlight, and form the world's very first towering forests.
When we trace this evolutionary timeline across deep time, the pace of innovation is just staggering. 1.2 billion years ago, we just have simple cyanobacteria. 470 million years ago, we start seeing the first sporal pollen-encoded spores of early land plants. By 425 million years ago, the first vascular plants with those internal transport systems appear in the fossil record. And then, just a geological blink later, by 385 million years ago, lignin-reinforced stems allow for the appearance of the first tall forests, which eventually culminates in the massive swampy forests of the Carboniferous period.
And here is a frankly mind-blowing stat about those ancient Carboniferous forests. Today, human beings still burn approximately 6 billion tons of coal every single year. But what is that coal really? It is actually the fossilized, undecayed peat from those ancient lignified vascular forests. Because the organic material in those massive swamps didn't fully decay back then, heat and pressure over millions of years essentially baked that early plant biochemistry directly into the coal we use to power our industrial modern world today.
Which leaves us with this incredibly profound and provocative paradox to end our explainer today. Think about it. The very same vascular plants that pulled carbon dioxide out of the Ordovician and Carboniferous atmospheres, causing massive global cooling and glaciations millions of years ago, are now the exact same coal that is rapidly returning that carbon to the atmosphere, warming our modern world. As we continue to unlock the complex molecular secrets of plant biology, we have to deeply consider, how will the biosphere react this time? Thank you for exploring the Green Blueprint with me. Keep analyzing the data and stay curious.